Hi, my name is Choi, and I conducted research on the efficacy of various bacteria in breaking down microplastics in the environment. Microplastics are pieces of plastic that are less than 5 millimeters and they are everywhere in our environment. Unfortunately, microplastics will only become more abundant as they are deposited directly into the environment every day from products such as face wash and laundry detergent. Additionally, larger pieces of plastic waste break down into microplastics. The most common types of microplastics in the environment are polyethylene, propylene, and polystyrene, which can be found in products ranging from food packaging to toys to synthetic cloth. Microplastics severely damage the marine ecosystem as they directly affect both the top and bottom of the food chain. Microplastics harm marine organisms on the bottom of the food chain, such as phytoplankton. Phytoplankton produce approximately 80% of the world's oxygen, but their ability to perform photosynthesis is inhibited by microplastics, which block the sunlight. Microplastics in the ocean result in a decrease in the amount of oxygen produced, which means that there is less oxygen in the ocean, which leads to dead zones. Dead zones are sections of the ocean where there is not enough oxygen to support sea life, leading to a decrease in biodiversity. The blue and red areas on this map indicate areas in the ocean where there are already low oxygen levels. Microplastics also directly affect organisms near the top of the food chain, including fish and seabirds, by blocking their digestive tract, causing them to die. Luckily, some types of bacteria, including Bacillus subtilis and Vibrio angularum, can degrade microplastics. Thus, this experiment sought to find which type of bacteria could degrade microplastics the fastest, as this is a very pressing issue that needs to be remedied. My specific research question was, which type of bacteria, Vibrio angularum or Bacillus subtilis, is able to degrade microplastics more efficiently and completely? And my hypothesis was that Vibrio angularum will be able to degrade microplastics more efficiently than Bacillus subtilis. I hypothesized that Vibrio angularum would be able to degrade microplastics more efficiently because in previous experiments it had been successful at degrading microplastics without any treatment. Bacillus subtilis, however, was only used in one previous experiment and it was first given a UV radiation treatment. As this sort of treatment would not be feasible in a real-world application, I chose not to use any treatments on the bacteria. As Vibrio angularum had already been successful in degrading microplastics without a treatment, I hypothesized that it would be able to degrade microplastics more efficiently than Bacillus subtilis. To conduct this experiment, I kept the Vibrio angularum in an incubator at 25 degrees Celsius and the Bacillus subtilis in an incubator at 30 degrees Celsius. The bacteria were plated onto agar plates as seen in figures 5 and 6. After 24 hours, the bacteria had grown enough to add microplastics. The microplastics were obtained from cutting open a parcel laundry pod and separating the detergent from the microplastics using mesh, as seen in figure 4. These microplastics were polyethylene terephthalate and polypropylene terephthalate. The microplastics were dyed blue and added to the bacteria using a metal spatula, as seen in figures 7 and 9. I dyed these microplastics blue because in a previous experiment the researchers were able to make their microplastics glow, but that wasn't something that I was going to be able to do in my lab at school. However, I still needed a way to make these microplastics visible, so I chose to dye them blue. I also used other microplastics that were larger and thus more easily visible. These microplastics were nylon flock fibers, figure 11, polyester glitter fragments, figure 12, polypropylene fibers, figure 13, and XAD2 polystyrene beads, figure 14. The petri dishes were observed under a microscope and photographed over the next 2 days. After 48 hours, the remaining microplastics were observed. Using a one-way ANOVA test, a statistically significant p-value was found for polyethylene terephthalate and polypropylene terephthalate, which was 6.31 times 10 to the negative 10th, nylon flock fibers, which was 7.24 times 10 to the negative 4th, and polypropylene fibers, which was 1.54 times 10 to the negative 5th. The p-value was not statistically significant for the XAD2 polystyrene beads, 0.329, or the polyester glitter fragments, which was 0.325. The Vibrio angularum was able to degrade microplastics more efficiently and completely within 48 hours. A statistically significant p-value was found for polyethylene terephthalate and polypropylene terephthalate, nylon flock fibers, and polypropylene fibers. The p-value was not statistically significant for the XAD2 polystyrene beads or the polyester glitter fragments. Overall, Vibrio angularum degrades microplastics more efficiently than Bacillus subtilis. These results mean that Vibrio angularum is a viable option to solving the microplastics problem. Vibrio angularum could be used in water treatment facilities to remove microplastics from the water before it's dumped into rivers, which then flow into the ocean. This would help protect the marine ecosystem from microplastics. Future experiments could use other types of bacteria that are known to degrade microplastics, such as Rhodocus ruber or Arthropacter globiformis, to test if these bacteria are able to degrade microplastics more efficiently than Vibrio angularum. Another future experiment could use other types of microplastics, such as high-density polyethylene, polyvinyl chloride, and low-density polyethylene, to test if Vibrio angularum and Bacillus subtilis are able to degrade these microplastics, and if Vibrio angularum is also able to degrade these microplastics as completely and efficiently. These are the references I used. Thanks for watching.